Hello there, ninth graders. This is teacher Krishna, and welcome to our biology class for week number 20. Thank you a lot for all of the efforts you have done for completing all of the online lessons that we have prepared for you, as well as all of the online assignments. Just like every week, log into Agilex, enter the Google Classroom, download the Word document from Google Classroom, and save it inside your biology folder, just like it is specified right below. As we move, remember that it's very important that you take notes so you can keep track of the most essential things from this presentation and video. And this is the ending mind that we have for today. We're going to study topic 3.05, and it goes like this. Students will be able to retell historical facts about the discovery of DNA, replication, as well as generalities on the DNA structure and its replication process. Alright, so make sure to keep track of this part in your Word document. So, we're going to start talking a little bit about DNA replication, of course relating it to the genetic code. So, we have one word which is very important, which is transcription, which is the synthesis of a RNA molecule from the DNA. So basically what RNA does is that it is going to copy information from the DNA that is in the nucleus. So once the RNA copied the information from the DNA in the nucleus, it is going to go out and it is going to enter the cytoplasm. Then we have a translation, which is the interpretation of the messenger RNA to attach a sequence of amino acids and create a protein. So once you already have the message, let's say, because it's messenger RNA, the one that copies the information from the nucleus, and it enters the cytoplasm, right there, and it is going to interact with some other components. And based on that information, a lot of amino acids are going to be put together, and they are going to create a new protein. There is another important term that we need to analyze, which is codon. It is a set of 3 nitrogenous bases in messenger RNA that codes a specific amino acid. It is the equivalent of a word in our alphabet. Alright, so remember that we had 4 different nitrogenous bases in DNA, which are adenine, thymine, guanine, and cytosine. Then in RNA we have 3 different, we have one of them that is different, in which we change a thymine for uracil. But when we say that we're analyzing a codon, it means that we are analyzing a set of 3 nitrogenous bases. For example, adenine, and uracil, or cytosine, and guanine. So those sets are codons. There are codons that signalize when to start and when to stop. That is very important because whenever the transfer RNA and the ribose are analyzing the information from the messenger RNA, they need to know when to start and when to finish. So we're going to analyze that a little bit more later on. So here you go. So these are all of the possible combinations that you can get in the codons that we were just mentioning before. So this one that is right here, that is signalized with an orange color, is the start codon. And you are going to find it whenever you find the combination of AUG. So when transfer RNA and ribose are reading the code and they read the AUG, then they are going to know that that is when they are going to start codificating all of the code. Alright, and how do they know when to stop? Well, they are going to know when to stop when they find any of these 3 codons that we have right here. So we have UAA, UAG, or UGA. Those are the stopping codons, okay? So after it has started already coding all of the information from the messenger RNA, they are going to start reading the information. For example, they find a codon that is CCU. Alright, so let's find that right around here, and it's right here, CCU. Okay, so this is the first amino acid that is going to be found. Okay, so CCU will be proline. And let's say that after CCU there is another codon with the letters AGU. Okay, let's find it around here. Okay, here it is, AGU. So the following one will be serine. Okay, so what happens to these amino acids that we have right here? Well, they are going to be put together. So the first amino acid that we are going to find is going to be proline. Then we are going to find serine, and so on and so forth. Let's say that right after, and there is another codification of GGC, so glycine is going to be sticked to serine. Then we find another one that is AAA, which stands for lysine. So then lysine is going to be stick to glycine. And let's suppose that then we find UAA. UAA, that is the stop codon. So one more time, that is the signal for transfer RNA and for the ribose to stop coding everything. Alright, so that is like a small example of how the amino acids are going to be put together. Let's continue. Translation. So after the transcription, a messenger RNA enters the cytoplasm. It interacts with transfer RNA and ribose. Just like we have been studying so far, the ribose attaches it to a messenger RNA. So the ribose is very important because remember that we mentioned the amino acids just before? So the ribose is in charge of putting all of the amino acids together. Okay? And transfer RNA has a set of 3 nitrogenous bases called anticodon. They match the ones in the messenger RNA and attach amino acids to form a new protein. So remember that the nitrogenous bases are always going to pair up. And if we talk about messenger RNA and transfer RNA, adenine is always going to pair up with uracil. And cytosine is always going to pair up with guanine. So basically, the transfer RNA has this anticodon, which is basically a group of 3 nitrogenous bases that are going to match the ones that it finds in the messenger RNA. So based on that, it is going to add a certain amino acid, and the ribose is going to be in charge of keep on putting all of the amino acids together to form a protein. The ribose attaches the amino acids until the translation reaches the stopping codon. We already analyzed what the stopping codons were. Alright, so here you have an example of what all of this process looks like. That you have right here, right at the bottom. This one is the messenger RNA. So all of these letters that you see right here are basically the information that it copied from the nucleus. Alright? Now this brownish thing that you can see right here is the ribose, and as you can notice, it is attached to the messenger RNA. And one more time, it is going to be putting together all of the amino acids that are coming right here. And lastly, the one that you find right over here, right at the top, this one will be the transfer RNA. And just like you can see, right here it has the anticodons that are matching the information from the messenger RNA. And based on what information it finds, it is going to add a certain amino acid that, one more time, is put together by the ribose with the other amino acids that are coming. And all of this process here is the process of the protein synthesis, for you to keep it in mind. Let's go pretty quickly over your evaluation. So read and summarize pages 6 to 9 from the topic we are studying today. Download the assignment on page number 9 of topic 3.05. Complete the report and upload it to the evaluation for topic 3.05. Check the rubric for evaluation in page 9 of topic 3.05. So, anyways, during the Zoom session, we're going to be discussing about this specific assignment, so you can understand a little bit better on how to complete it, because sometimes it might be a little bit tricky. Of course, I want you to try it out by yourselves, but if you're doubting, you can let me know in the Zoom session so I can help you out. That was it for today's session, guys. It's been a pleasure to have you here, and I will see you just in a couple of minutes. Goodbye.